Good afternoon. My name is Barbara McMakin, and I'm from the NIN's Office of Neuroscience Communications and Engagement. On behalf of the NIH, I would like to welcome you to this afternoon's call and to thank you for your interest in participating in this discussion with us today. Today's call is being recorded. If you have any objections, please disconnect at this time. Dr. Walter Koroshetz, Director of NINDS, will introduce the speakers, each of whom will make some remarks, after which we will answer your questions. If you have a question for our speakers, we invite you to submit it through the Q&A box at the bottom of the Zoom screen. We will try to make our remarks brief so we can answer as many questions as possible in the time we have available to us this afternoon. Now, Dr. Koroshetz.
Good afternoon and welcome to the second MECFS telebriefing of 2021. Since our last call, there have been several developments at NIH related to MECFS, and we look forward to updating you on what we have been doing. Unfortunately, we are still dealing with the pandemic, and many of the clinical studies are still not quite up to speed, but certainly doing better than they were a couple of months ago. And as you know, there's considerable evidence that people will be suffering from MECFS after they've passed through the acute infection stage. And there's a great deal of science that's being launched to try and solve that problem best we can, given the emergency situation that has occurred across the country. And we'll talk a little bit later as we think that this work has real potential for uncovering some of the biological drivers underlying MECFS in general terms. As you may know, a number of years ago, NIH, many different institutes, developed a consortium for MECFS collaborative centers and a data management center. They're coming into their 5th year, and all the NIH grants are pretty much 5-year grants. But the councils at NINDS and we have input from the other institutes as well, have approved reissuing the funding announcement to support the MECFS collaborative centers. And this would be support for an additional 5 years. And we expect that these funding announcements will be released in the next few months. And Dr. Breen will talk to you a little bit later about a very interesting meeting that the consortium members held recently. Similarly, during the call today, you'll hear from Dr. Nath, who will provide updates on the intramural MECFS study. As you know, Dr. Nath is the clinical director of NINDS intramural program. Dr. Joe Breen from NIAID will let us know what has been happening in the consortium and about the recent annual meeting. And then Dr. Vicki Whittemore will discuss activities of the Trans NIH MECFS Working Group and a very interesting recent workshop that was held with the Sleep Research Society on the biology of fatigue. In addition, we're really excited. We have a guest speaker this afternoon, Dr. Rakib Rehan from Georgetown University, and he'll be giving a brief presentation. But to know that we have made major efforts to try to attract young scientists into the MECFS field, and he's the first to receive what we call the F30 Training Grant Award that NIH has, and he received this award to investigate the neural underpinnings of chronic fatigue. So we're real excited to have new young investigator join the fold. As always, we are eager to hear from you, so we will keep our own remarks very brief, and then we'll be turning to the questions that occur in the Q&A box. With that, let me turn it over to Dr. Avi Nath.
Thank you, Dr. Koroshetz. So I'm going to give you a brief update on what's been happening with the intramural program. as you know, that when the COVID pandemic hit, we moved into data analysis mode. And so at that time, we had 17 ME/CFS patients that had been fully adjudicated and 21 healthy controls. So we formed several working groups. We had a clinical description working group, an immunology working group, and physiology working group. And so because it's a trans-NIH effort, so there were lots of people within each of these working groups. They have spent a numerous amount of time collecting and analyzing all the data, and then going back and doing additional experiments as necessary to confirm some findings. And so most of that data is now been fully analyzed. There's some few remaining things that still need to be done. So now we have formed a manuscript writing group. So that consists of the leaders of each of these working groups and a couple additional people. And so we are now starting to put together the figures and the rest of it. So we hope that we will be able to produce a substantial manuscript with all the findings that we have in short period of time. The major observations there are that we have found a number of physiological abnormalities in the testing that we've done, as well as immunological abnormalities. The challenge is to really synthesize it into a single story, which seems to be a bit of a challenge, but I think that's the challenge within the field. It will point in the direction for what additional studies need to be done in other cohorts, and may even point to potential therapeutic targets that would lead to a subsequent study. In the meantime, what we have done is, since a lot of the COVID patients now have symptoms that are overlapped with ME/CFS and may very well be entirely the same disease, we have established a huge cohort of those patients. Another 400 patients we have screened, and we have several protocols now, both for phenotyping as well as for intervention. And so we're going to be bringing those patients now to the clinical center for further study. So this will continue with the slightly different population, but the questions are still the same. There's another study that Dr. Wallet is now the PI of, and that is the Gulf War syndrome, in collaboration with the VA system. And that has been funded just within the last few days. So there'll be another cohort of patients coming in, but the questions are still the same. And I think the benefits will be for the greater community of ME/CSF from all these studies put together. I'll stop here. Thank you.
Thank you, Avi. And now we'll turn to Dr. Joe Breen to talk to us about the consortium and the recent consortium meeting.
Thank you, Dr. Koroshetz. My name is Joe Breen. I'm the National Institute of Allergy and Infectious Diseases representative on the NIH working group. Work very closely with Dr. Koroshetz and others on the call here. And I'm going to just briefly tell you a little bit about the annual meeting for the collaborative research centers that Dr. Koroshetz mentioned are entering into their final year of support. It was held just about a week ago, and representatives from all the centers, as well as our close Canadian collaborators, participated. And notably, Community Advisory Committee involvement was also notable. I'll say a little bit more about that in a second. So there was, I think, a lot of convergence on data presented. A lot of the data is pre-publication, but I think it will be published quite soon. And I think the feeling from the meeting was that there was a lot of convergence on some findings, namely metabolomics and some microbiome biomarker type or differences that were seen in ME/CFS patients versus controls. And this was exciting because some of the very similar changes were seen in different experimental settings and with different methodologies, in fact. So this is one of the goals of the centers was at the outset to try and validate findings across centers. And that is something that we're starting to see achieved, even given the issues with recruiting patients, et cetera, with the pandemic. And most of the centers are able to now recruit again, but there was an obvious lag, unfortunately. But there was some really nice enthusiasm, some strong correlations and associations that were noted. So I mentioned that there was input at the meeting from the Community Advisory Committee, and their input will be publicly available quite soon. And that would be in the form of feedback from the meeting itself, as well as suggestions about feasibility of further studies and priorities based on their input. So that was welcome. And the Data Management Coordinating Center presented some ideas about how to further integrate and improve some very valuable products that they've generated called MAP MECFS for a data sharing, and as well as Search MECFS, a way to access samples that could be accessible really, again, for discovery, but also cross-validation. So we're looking forward to formal input from the Community Advisory Committee. And the other thing, from the results, I mentioned the metabolomics and microbiota, but there was also some convergence around age and gender differences were important in terms of patient population to look for markers. So it would be really exciting if this was to be validated, and again, since multiple groups are seeing some of these common changes, and they would tending to be in energy metabolism, some really classic energy metabolism pathways that in fact are well studied. And if they turn out to be more than associative here, there may be ways to disrupt those pathways. So from my perspective, I think there was some really positive outcome, some nice productivity which will be represented it is represented in some publications, and I think we'll see more. And I'll stop there and happy to answer any questions when we get to that part. Back to you, Dr. Korshett.
Thanks, Joe. Now let me turn to Dr. Whittemore to talk to us about the Trans NIH working group for MECFS and the recent workshop on the neurobiology of fatigue.
Yeah, thank you, Dr. Koroshetz. So the Trans NIH MECFS working group has been focused quite a lot on the reissuing of the RFAs for the centers and for the Data Management Coordinating Center, as well as thinking about strategic planning and how to move that process forward on both the NIH side and on the extramural side. So investigators and clinicians became very swamped with all of the long COVID work. Many of us on the NIH side were pulled into working on working groups for the Recover initiatives. And when we discussed starting a strategic planning process with investigators and clinicians, they said, many of them said, we simply don't have time. But we have plans in place to begin to move that forward. And we actually have started to have conversations with the FDA and CDC around clinical endpoints for clinical trials, as well as conversations with both US and European clinicians to really think about harmonizing clinical trial endpoints so that we can really move that area of research forward toward clinical trials. And I think some of our initial conversations are going to really lead to pointing out the need for some very specific targeted research to validate and to understand how to utilize some of the tools that we have out there in better ways as we move forward in clinical studies and clinical trials. So just to touch quickly on the fatigue workshop, this was a workshop that, as what Dr. Korshett said, was organized together with a committee, a working group from NIH, made up of individuals, many of them who also are on the Trans NIH working group, but who made up a working group as part of the Blueprint Neuroscience Research Program at NIH. And together we partnered with individuals from the Sleep Research Society. And the goal of the research of the workshop really was to bring researchers together to talk about how is fatigue described and studied across different diseases, not just chronic fatigue syndrome, but we touched on MS, on autoimmune disease, on cancer, on other types of diseases, and then to think about the potential underlying mechanisms of fatigue. How is it being studied both in clinical populations as well as animal models? And then the last session really focused on the neurobiology of fatigue and what do we understand about how we perceive fatigue, how is potentially cerebral spinal fluid flow and sleep cycles related to fatigue and cognitive dysfunction. And with the emphasis from the Sleep Research Society focus is what is the role of sleep and unrefreshing sleep, and the role that plays in really fostering chronic fatigue within many different syndromes. And we had actually several individuals who are ME/CFS investigators who participated in the workshop. And the next steps from that is we're working very quickly to put out a report and summary from that, and internally talking about what are the next steps to really foster cross-disciplinary, multidisciplinary research on fatigue that I think will really help us much more broadly across diseases to understand what are the underlying mechanisms, how can we target those mechanisms and develop treatments that will really help to alleviate and hopefully eliminate fatigue in its chronic stages especially. So with that, I will stop and turn it back to Dr. Koroshetz.
Well, thank you very much, Vicki. And let's see. There is the biology of fatigue workshop. I think is the video casts are now available, I think, for people to see. Not sure how, maybe someone can put the link in the Q&A so others can see it.
Yeah, it's on the Blueprint website, and we'll put that link in the Q&A.
Great. Okay, well, thanks very much. And I think Dr. Rahan was in clinic, if I remember right, so I'm not sure if he was able to join yet. Barbara, do you know? Not that
no, he's not on yet.
Okay, before we go to the questions, there was, I did receive a couple in advance. There we go. So there were, there were a number of questions about the Recover Initiative and the interplay with ME/CFS. So I think the main points are that the significant symptom overlap between what people are suffering from, what's called long COVID, and what people are suffering from in ME/CFS. And in the beginning, the only real difference was that the duration wasn't qualifying for ME/CFS, but that has passed. Now I think a large proportion of people who are suffering from long COVID are now out to the 6 months, and they have fatigue, they have cognitive trouble, they have autonomic trouble like postural orthostatic tachycardia, they have unrefreshing sleep, and some of them have post-exertional malaise. So they are going to qualify for what we call ME/CFS. I think that's that's an important thing to understand. And with regard to that, as I've said in other instances, that this terrible pandemic does actually present a unique opportunity to understand these symptoms and those overlapping with ME/CFS in a way in which we could never do before. And that is because in the case of ME/CFS, many folks dated their symptoms to a particular event which sounded like an infectious illness, a viral illness, but we didn't know what the virus is. People came in months, years afterwards, and although we've tried, it's been very difficult to try to understand these biological underpinnings. But with COVID, we have thousands, tens of thousands of people coming out. We know what the virus is. We know exactly when they got infected. And we have the ability to actually study people as they recover from the virus to understand why some recover quickly and others have persistent symptoms. And that's something we could never do before in our ME/CFS research. Now, people have asked, well, why don't you do the same thing for ME/CFS people that you're doing for the recovered people? And the fact is that you can't, because you don't have that opportunity to see people who are acutely infected. And so that comparison of those who are getting better quickly versus those who are not getting better quickly, it's really only in the case of COVID can you do that. And similarly, if you look at people who have persistent symptoms at 6 months or a year afterwards, the control group is people who had COVID 6 or 7 months ago matched. And that same group is not possible with ME/CFS because those folks have usually had disease for many years. So it's comparing apples and oranges. What I do think is going to happen, and this is where I'm very hopeful, that we will have some findings coming out of the Recover Initiative looking at the recovery from COVID. And as soon as those are nailed down, then we can look to see if they generalize to an ME/CFS group that was not affected by COVID. So I think that is kind of the most beneficial way of looking at this. Try and milk everything that comes out of the post-COVID to see if it applies to ME/CFS. So I hope that helps a little bit in clearing up these issues. Now, and the Recover program is by Congress, 100% devoted to COVID. So that's not, we can't use that money for other purposes. And with regard to knowing who in the Recover cohort has symptoms overlapping with ME/CFS, I think that Vicki and Joe and a big team worked with the people setting up the common data elements. So fatigue, cognition, posturastatic tachycardia, unrefreshing sleep, and post-associative malaise elements are going to be collected on every individual who comes into that cohort. And the cohort, we're looking for 15,000 people and a control group, those who are infected, those who have persistent symptoms, 6 months later, those who are 6 months out and no symptoms. You also have an autopsy program, which will look at people who have died, who have post-long COVID. They probably did not die from long COVID. They probably died from other causes, but it'll give us an opportunity, which we've never really been able to do in ME/CFS, is to look deeply into the tissues of the body for signs of immunologic or viral pathology. So again, that's something that could potentially shortcut to an answer. So, and also I think that we have, certainly right from the beginning, had investigators who are sophisticated in ME/CFS as part of the groups that inform the Recover Initiative. There are hundreds of investigators in this project, really hundreds. I suspect that there are people there who know something about ME/CFS, but also I'm very hopeful that of those hundreds of people in Recover, that because of the similarities, they will actually become ME/CFS researchers in the future as well. I just wanted to kind of address those issues with regard to the Recover initiative straight off. Now, I see Dr. Rahan has been able to join. Is that right? Akib, are you good? Oh, he's in the
Yeah,
call.
can you hear me?
Yeah.
So I'm working at the hospital right now, and the entire hospital's like Wi-Fi is done. So it's now trying to figure out. Yeah, no, it's okay. It's okay.
Okay, well, I think people would really love to hear about your project, and then also, how did you get into it in the first place, and how can we get people into it, this area of science? So, yeah, what could you offer folks?
Well, I can just start. I think because of all the technical difficulties, I think Miss Barbara has like my slides, and so if she is on, she can hopefully maybe put them up. Yeah, just a couple of slides here and there. That way we can.
Cool. Very good.
Yeah. So just briefly about me, my name is Ricky Rehan. I am an MD PhD, and I completed my MD PhD at Howard University College of Medicine in Washington, D.C. And completed very recently, and now currently I'm a first-year resident in the Department of Neurology at Georgetown University Hospital. So it's my first year, intern year, and right now I'm doing a rotation in the VA, and unfortunately everything in the VA, their Wi-Fi is not working. So at this point, I thought, let's go to my car. But a little bit more about me. When it came to studying and going into fatigue research, I actually started a long time ago, 2010, 2011. I was a research assistant in Dr. James Baranick's laboratory in Georgetown University Medical Center, and really fell in love with research, fell in love with neuroimaging, and trying to really understand kind of the underpinnings of fatigue, something that we see often in a clinical setting, pretty much in any chronic illness, but it's pretty much ignored. And so since then, it's been kind of my research track. So today I had this wonderful opportunity from Dr. Cotone to be able to present the work that I've done. This is specifically geared towards the grant that I got during my predoctoral stage, studying fatigue and using neuroimaging to study fatigue. This was an F30 grant. So the title of my thesis work for this was investigating the neural underpinnings of chronic fatigue. And as you can see, a multimodal approach. You can go to the next slide. And that's a brief overview we just went over. We can also, so just about the F30 grant, the initial application was in 2017, and the proposal was really kind of taking advantage of something that we saw in another fatiguing population called Gulf War illness, where we were able to use exercise and fMRI to kind of see what the brain does before and after some sort of exertional exhaustion, something that kind of models exertional exhaustion. From those findings in the Gulf War illness study, we proposed a similar type of study in CFS. And the rationale was because of the similarities in the symptomatology, we were expecting to see similar findings as far as brain patterns go. So next slide. That's the rationale again is, as we've talked about, when it's fatigue is, very subjective, part of pretty much any chronic illness, but despite its significance, we really just don't know much about it, as we all know. There's really no strong consensus as far as a mechanism or an underlying etiology that really kind of leads to the chronicity of fatigue. And so we were hoping that taking this kind of exercise modality, we could maybe kind of standardize a way of studying fatigue, taking away maybe leaning less on maybe 0 to 10 rating scales and more so on something that we can quantify. So next slide. And so one of the big things that I proposed in this grant or this proposal was taking advantage of the cardinal complaint, as we all know, post-exertional malaise. It was like really intriguing to me as a symptom, particularly because of the fact of how long it is and how and how debilitating that symptomatology is for our patients. And so by looking at it in a standardized way, saying, hey, you know what, let's look at the brain and how it activates before some sort of exercise, and look at how the brain activates afterwards, maybe we can identify why patients have this prolonged malaise in ME/CFS after some sort of exertion. And it really did present that wonderful opportunity because we were able to kind of standardize things. Oftentimes when I see patients with ME/CFS, many have described, the duration of their symptoms as being variable. Some may say for 10 years, some come in maybe 3-4 years, 20 years. And so by using this type of modality, we were able to kind of say, okay, you know what, we have this very discrete amount of time, and we can now look at everyone maybe in more a homogeneous way, and maybe get some sort of great data out of it. So next slide. So this is just kind of the brief overview of what we call, or I love when we call, the sandwich protocol. Patients were stuck into an MRI before any exercises. Then we put them on a bike, exercise them. And then after those 2 bicycle exercise stress tests, we took another scan of their brain. And we compared what the brain looked like before and after. We did that with controls, and obviously both were illness. And then for the specific F30, the grant that I was working on, we did it with ME/CFS patients, quite a large number actually for an fMRI study. Next slide. So just briefly, this is what we saw in our GWI, our initial study, was that, before exercise, we had the controls, and we had these 2 subgroups of GWI that we really won't go into. And then after exercise, what we saw was that those 2 GWI subgroups of the other fatiguing illness had this really increased amount of activity within this region that we know as the default mode network, which was really intriguing. It was really exciting to see. Nothing like that has ever been reported before. And so we thought maybe something like this could be seen in ME/CFS. Next slide. So what is the default mode network? It is actually, to me at least, I would consider it to be the core, one of the core, if not the, just linchpin network within the brain. It interacts with pretty much every other network within the brain. It modulates every other network within the brain. And in fact, even slight disruptions within this network has been seen across many myriad of diseases, whether you can say depression, schizophrenia, Alzheimer's, Parkinson's, any sort of debilitating cognitive illness that you can think of often has some sort of disruption within the default mode network. Next slide. So here what we did was, looking at the methods and just the number of controls and ME/CFS that we had, we were able to recruit, a pretty large number. I believe for the entire study, we had about 57, close to 60 people, which is actually pretty big for a MRI study. And we were able to have pretty, I don't want to say equal groups, but pretty, as far as ages go, they were pretty equal for that. And what we saw again, fortunately, was that after exercise, ME/CFS patients did have discrete changes within their brain patterns. Go to the next slide. And so this is just kind of the brief methods. The methods that we used here is that we looked at the functional activity within the brains, and we broke them down into nodes. And these nodes are parcellations of the networks that we have within the brain. And then once we parcellated it out, we used kind of machine learning, hierarchical clustering to be able to kind of make identifications of these networks. And so what we saw is that if you look all the way to the left, the purple, I'm not sure if you can see it, that area right there is the default mode network, or the subnodes, as we say, which includes the medial prefrontal cortex, the precuneus, and parts of the parietal as well. Go to the next slide. So what we saw was in our ME/CFS patients was, before exercise, again, something is that there was really no significant difference within the activation patterns within our controls versus our ME/CFS patients. However, after exercise, what we saw was there was a significant increase in activity within the medial prefrontal cortex. And so that was something that we were excited about, because not only did it give us, a reproducibility as far as our exercise protocol goes, but it also showed to us that the default mode network indeed does play a role within these fatiguing illnesses. Next slide. The other important thing was we were able to also see that the activation within these discrete regions also correlated with the severity of the symptoms, particularly after exercise. Prior to exercise, the symptoms did not correlate with the activity, but after exercise, when we looked at the activation patterns and we correlated to the symptoms, we saw that there was actually a significant correlation. So that was something that was also very exciting for us, because it shows that you're going to have to stress the system before you're able to really see some sort of actual pathophysiology within ME/CFS. Next slide. So really what it comes down to is, I've been kind of condensing almost 3-4 years of research in about 10 minutes, but what we saw was that the medial prefrontal cortex itself has a very important role, not only within the default mode network, but when it comes to the subjective appraisal and awareness of fatigue. In fact, the medial prefrontal cortex is one of the first regions to start to activate when someone is doing a fatiguing task. They've shown in multiple studies that if you do a fatiguing task of some sort, the medial prefrontal cortex is one of the first regions to start to activate. In fact, if you have disease within that region, let's say from a TBI, those patients tend to have more levels of fatigue than patients who have TBI and in other regions of the brain. So the implications of all the research that I was able to do was, one, we were able to reproduce a model, something that was standardized. We didn't have to rely on ratings anymore. And two, we were able to kind of show that MRI, a non-invasive method, really has some utility here. And finally, one of the big things is maybe, hopefully, attempting to find potential pharmacological targets here. So next slide. So the summary of all the work that we did was, again, we identified a model. We were able to study and detect fatigue objectively without ratings of any sort. We were able to really see how to modeling post-exertional malaise in 2 separate groups using this modality was robust enough to be able to really find great fMRI findings. And so the evidence itself, one of the takeaway points that we can say here is that the inability to stop the default mode network from activating could possibly be a potential biomarker for fatigue. It could be that that region or that network is just not appropriately controlled, maybe in ME/CFS or other fatiguing illnesses. And so all of this work that I just went over is going to be published soon. The final manuscript for all of this is in the late stages of review. And we can go on to the next slide. And so really now comes the future direction. So often the question is, well, so what? What does this all mean? You did all this work for maybe a couple of years. Can you tell us what your next steps are? One, prior to undertaking any of this type of research, we knew that the brain had, some sort of involvement or role within the chronicity of fatigue. But after this study, we were able to kind of parcelate and kind of identify discrete regions now, which is really important. The brain has multiple regions, hundreds of regions, in fact, that have distinct functional components to it. And to be able to now say that, we have 3 or 4 regions that are within the default mode network that are associated with fatigue, it kind of now gives us a way to pinpoint or figure out a better way to obtain the next research step. So at least for me, from a research perspective, the next steps would be to do something called pharmacological neuroimaging. Can we look at before and after? Can we find targets for these regions? And so there has been studies that have shown that several serotonin receptors and opioid receptors, even glutamate receptors, are significantly associated or with these regions versus other regions, and those could be potential targets in the future. And so just thinking about that, it's really exciting just because nowadays, ideally you would want, pharmacological therapy to start in the murine models, but because of the fact that we have known targets, we have known medications that can go to these targets, it gives us an opportunity to really kind of push the translational foot forward here. So that's the hope as for the next steps as far as research goes with what we're doing. The last slide is just like a thank you or like maybe a bibliography. Yeah, it's just, boatload of bibliography. But that is about 5 years of research in about 5 to 10 minutes. And more than happy to take any questions.
Yeah, well, thanks so much. Very interesting data. So if I just simplify it, the finding was increased activity after exercise in prefrontal cortex.
Mm-hmm.
Okay, and then so where any thoughts on where the drivers are? Is there a decrease in inhibition of prefrontal cortex that's coming from somewhere, or an increase in excitatory input into prefrontal cortex?
It could be one or the other.
What's upstream? What's upstream? Yeah.
Yeah, I think it could be one or the other. I think that's why the pharmacological targets and having next step research that takes advantage of that will maybe allow us to better answer that question. I think if we know that it's an either inhibitory or excitatory issue, at least from what I've seen when it comes to the most cutting edge research of receptor within the brain, I think this would be more so of a loss of inhibition versus a gain in excitatory activity. So I think that's where I'm leaning, but I definitely would need to research that before we can make that as a solid claim.
Yeah, interesting. Yeah, there are some studies in the motor cortex with fatigue that suggest that the motor cortex is actually inhibited in the time of fatigue.
Mm-hmm.
You can get a strong motor output by transcranial stimulation in the fatigue state, but not in the non-fatigue state. So that, but again, prefrontal cortex is so different than M1s. But yeah, really interesting to try to piece these together. Certainly prefrontal cortex has a lot to do with the reward system.
Right.
I'm sure there's a lot of inputs from limbic cortex, amygdala.
Right.
Yeah.
I think one of the things is, at least from the data we've seen, is that the most significant activation patterns were more so in the lower parts, the ventral medial, much more closer to the limbic system. And so you could, at least from all the data that I've seen, if there is an increase of activation or neuronal activity within those lower regions, they would kind of crosstalk to the higher, more, higher level regions and kind of shut them off. So I'm thinking that could possibly be one of the reasons why, CFS or ME/CFS may have such a broad symptomatology, right? In the sense of brain fog and word loss or memory loss and things of that nature. I think it could be some sort of one point start and it affects all parts of the higher level parts of the brain. So, I mean, it's lots to still kind of dig into for sure.
Right. Yeah. Oh, I could talk to you all day about this. Very interesting. Really appreciate it. Really appreciate you coming on. Yeah.
Yeah. Do you have time to answer a couple questions, or do you need to get back into the hospital? You're
no,
okay.
No, I told them, like I told my other resident that just take care of the patients for now. And yeah, questions, I'm more than happy to answer. Anyone has
Right,
questions?
terrific. So there was a question, if you could just repeat sort of the protocol, the exercise protocol that you used in your study.
Sure. So the protocol itself, one, patients would come in, and prior to doing any exercise, we would get this baseline fMRI scan. And so within the scanner, patients would get something called a resting state scan, where they would just kind of sit there not doing anything. And then they would do actually a task as well in the scanner. It's called a working memory task. And within these functional realms, we would also get structural scans as well. So we would get white matter data, gray matter data. And then after all of that pre-exercise brain scan is done, they would then get on a bike. And so this wasn't a treadmill. It was we would call a submaximal exercise stress test. Dr. Brannick, I think, is somewhere on the call, but pretty much the gist of that exercise cycling test is that patients would kind of reach their heart rate up to 70%, and then, right to get to 85%, and then we would stop. We wouldn't attempt to try to push the patient to true exhaustion, just because we did have a second exercise coming up the next day. So it was enough to induce fatigue, but it wasn't enough to cause, I would say, just outright, completely, like, they wouldn't be able to do the test the next day or go into the scan the next day. I think those were kind of the important parts of it. But it was the protocol itself, the bicycle exercise protocol, at least from what we've seen from the data, was sufficient enough to induce post-exertional malaise, the objective findings that we did see on the MRI. So.
Yeah, thank you. So there were lots of comments in the Q&A thanking you for the work that you've done and that it's amazing. So thank you.
Thanks.
And sort of following on to that question, there's huge controversy in the field about the use of graded exercise in chronic fatigue syndrome. And there's a question about whether this brain imaging is actually a demonstration of the impact of exercise. It can actually show you the impact of exercise on fatigue and on individuals with ME/CFS.
Yeah, I mean, that was kind of the goal, was to be able to have controls and ME/CFS patients do a standardized exercise protocol. I think it's not the exercise that we were focusing on. It was more so of having patients go into an induced fatigue state or be part of something that causes exertion, where the body's systems are feeling stressed. And I think if there could be several things that we could have used, but I think this was kind of the best way to approach a standardized way of inducing exertion within a large population. And the graded exercise controversies, I know it's back and forth, but I think at least for us within this protocol, the attempt was to induce a fatigue state acutely, and I think that's what we were trying to really achieve here. So yeah.
And there's several questions that sort of get at how does a viral infection, neuroinflammation potentially lead to down the road showing these kinds of results in the brain using this kind of imaging? Sort of what's that connection of someone who's now post-infection having these kinds of results? Can you make those connections or not?
Though I unfortunately don't think you could, just because there are so many insults that could occur to the brain and to the brain's milieu, as we would say, throughout maybe their initial viral infection to when they finally were maybe diagnosed with an ME/CFS. It'd be very hard to say specifically. It would be very difficult to say. Yeah. But obviously, there is associations with, post-viral illnesses, obviously with COVID now, and even before COVID, we've had that association in ME/CFS. But as far as the mechanisms go, it just would be very hard to say at this point.
Okay, yeah, there's lots of questions. Actually, I see a job offer here for you in the Q&A too.
I'm sorry?
I see a job offer here asking if you'd like to come work at the Bateman Horn Center.
got to finish his residency first.
I know.
Yeah, so there's a question that just came in about if you could talk about patient selection methods and diagnostic inclusion criteria you used to select the individuals with ME/CFS who participated.
Sure. So the patients, just had to meet the criteria for ME/CFS. We really didn't, our exclusion criteria were really kind of focused on making sure that they didn't have like congestive heart failure or things that would kind of exclude them from being a part of an exercise study. Another exclusion we had was really, no metal implants or things that would, claustrophobia. But we really didn't have too many, I would say, exclusions that I would say would muck up the patient population. If you had ME/CFS, you met that criteria, and there wasn't, like I said, like heart failure or some sort of cardiac issues or inability to actually participate within the study, we had a pretty solid number from a wide group, I would say, of patients with ME/CFS. They didn't fit any specific cluster. So yeah, our exclusion criteria wasn't large.
Okay, and one last question for you before we move on.
Sure.
To get to some other questions here. Did you evaluate heart and lung issues with the participants while participants were on the bike? For example, did you test your VO2 max to see whether they
we did.
Going into anaerobic state?
Yeah, we did. That's all in the manuscripts, as my my mentor, Dr. Bernick, would say. But yeah, we did. We did before exercise. We also did serial measurements of orthostatics throughout the entire exercise protocol. And so there were several components of that. As far as the lung goes, the only time was, during the exercise, we did look at, pulmonary capacity, VO2 capacity, but we didn't do, let's say, like what do you call that? The blow tests. We didn't do those as settings. We did just the orthostatics with the as far as heart rate and blood pressure goes, but that's about it. We didn't do anything like that.
Okay, well, once again, thank you so much for joining us today and for the research you've done. And we hope you come back to ME/CFS research when you finish your training.
I will.
Excellent, thanks.
Thank you so much.
So we're going to move to a couple of other questions. There was a question about if the ME/CFS collaborative centers can receive funding through the Recover initiative. And I'll just answer that quickly in that the initial call for proposals were open to any institutions and investigators to submit applications. And those sites have, my understanding is, have not yet, but are soon to be announced. And there will hopefully be additional initiatives coming down the pike, because this first phase is to collect the biospecimens and clinical data from individuals with long COVID, acute COVID and long COVID, and then make those biospecimens and data available to investigators. So there will be further initiatives coming along down the road. I think the next question I'd like to direct to Dr. Koroshetz about back to your discussion about the appropriation of the funding and that it was for long COVID, and really the question of, do you think that these protocols and the sites will really be set up to identify individuals with ME/CFS in those cohorts, even though the funding is really directed to long COVID? So what do you think about that question?
Well, yeah, I think as I was mentioning, we and you were very helpful in getting the critical components of the symptoms of ME/CFS patients into the common data elements for the Recover initiative. So, Recover is going to look at all aspects post-COVID. Is the pancreas been damaged? Is the shortness of breath due to pulmonary fibrosis? In addition to issues, that we're real sensitive to, what causes the fatigue, the post-exertional malaise, sleep disorder, POTS. So those data will be captured, and patients, I suspect, will fall into groups. And what we know about long COVID now is that there's a list of up to 70 symptoms that people experience, and then there are clusters of symptoms. So we're looking at this from first principles and trying to put together, the biological drivers for these clusters. There may be more than one. There may be more than one thing going on. But I don't think there's going to be a problem identifying this group who actually fits the criteria that we know of as ME/CFS. And as I said, we're hopeful that this is an opportunity that you're not going to get again, hopefully, to understand what causes this. And then the question is, can it be, whatever we find, can it generalize to the ME/CFS population that was not COVID? So if everything's COVID specific, then this may not help so much. But I would guess that given the fact that many different post-infectious symptoms that become persistent are so similar, that there's going to be an overlap between the people who have long COVID with ME/CFS criteria and people who have ME/CFS criteria before COVID. And then in terms of clinical trials, I think, once again, you have this homogeneous population in COVID patients that we're hoping once we find something that we can actually try and test an intervention. And again, those interventions, if they work, would certainly be high on the list to try then in people who have ME/CFS unrelated to COVID. And then thirdly, as is in the chat many times, we were interested in funding more research in ME/CFS. And that has been, really tough because we don't have, we need 1,000 Rakibs, and he's actually our first one to get an F-30. So, but we have, I don't know, there might be 1,000 people investigated in recover, and they're going to be working with patients that have symptoms that are very similar to ME/CFS patients have. So I think that's another opportunity to draw from investigators who are, in this short time frame on COVID, but over the long run to get them really interested in studying ME/CFS in the generic space. So I think there's a lot of opportunities for us as interested in ME/CFS to draw things out of the recover. And again, it's an amazing, ambitious project with, tens of thousands of patients, hundreds or maybe 1,000 investigators. It's not at people say about NYU. It's not at NYU. That's just a coordinating center. It's all over the country. So I think we got to be hopeful. But this is a time to really press down on long COVID symptoms.
Yeah, thank you. And I'll just add that although the number of grant applications coming in, we're encouraged because we're being contacted by investigators from outside the ME/CFS research area who are interested in doing research on ME/CFS. So I've had several calls recently with new investigators who are interested in moving into this area. And, there's a question about the discrepancy, for example, between funding for MS, multiple sclerosis, and ME/CFS. And, I think one of the differences there is that the MS Society has been around for many, many years and has pumped millions of research dollars into seed grants, which have really us, and those investigators then were able to get grants from NIH. And we're beginning to see that with several investigators who had seed grants from SOLVE, ME/CFS initiative, who are now able to translate those preliminary findings into grants that are now being supported by NIH. So we have many more questions I wish we could get to, but we're at the end of the time, so I'll turn it back to Barbara.
Great, thank you so much, everyone. A recording and transcript of the call will be posted on the NIH MECFS website soon. And also, I'd like to remind everyone about our listserv for updates from NIH. To be added to the listserv, please visit the NIH MECFS website at www.nih.gov/MECFS and click on Join Our Listserv at the bottom of the left sidebar. Thank you again for today's call and have a great afternoon.
Thanks, everyone.